that's really what led after a few years to what I think I'm mostly known for in the field, which is pioneering the idea that repairing the molecular and cellular damage of aging is actually going to be easier than making the body run more cleanly and slowing down the rate at which the body generates that damage in the first place, which is where everybody else had been focused.
Hello, my name is José Navarro-Betancourt, and I am the scientific director of quadraScope, a venture capital fund that invests in early-stage biotechnology companies aiming to dramatically extend human health span by targeting the hallmarks of aging. The following is a conversation with Dr. Aubrey de Grey, a pioneer in the field of longevity. Over 20 years ago, Dr. de Grey was one of the first scientists to propose that aging can be treated or slowed down by fixing damage at the molecular and cellular levels. He is the scientific founder of the SENS Research Foundation, and he recently founded the Longevity Escape Velocity Foundation, which we cover in the interview. He also sits on the advisory boards of select journals and organizations, and we are lucky to have him as an advisor at quadraScope. It is also interesting to mention that he has a multidisciplinary background. He actually began his career as a computer scientist and later earned a PhD in biology from the University of Cambridge. Now, this is my conversation with Dr. Aubrey de Grey. Hello, Aubrey. Thank you very much for taking the time to do this interview with me.
Well, thank you for having me.
So, Aubrey, you have a very interesting background as a computer scientist. So how did you become involved in biology and then the longevity science and industry?
Yeah, I guess it's quite an unusual story. So yes, I understood from a very early age, like, my early teens, that what I wanted to do with my life was to make a big difference to the world, to work on some very serious problem for humanity.
Mm-hmm.
And I went into computer science because I found out when I was 15 that I was pretty good at programming. So I thought, okay, the thing I'm going to work on is artificial intelligence research, because fundamentally that's really all about automation. It's all about getting machines to do stuff that we would rather not do and that we only actually do because we're being paid for it. Um, so I did my undergraduate degree in computer science, and then I worked for 6 or 7 years on artificial intelligence research. The specific area that I was focused on was software verification, which is all about writing programs that find mistakes in other programs. And this was way, way back in the 1980s, before the huge revolution in machine learning and neural nets and so on that's happened over the past decade or so. Back then, everything was in the world of mathematical proofs and symbolic reasoning, and so it was very, very different from how things are now. But anyway, during that time, around 1990, I met and shortly afterwards married a biologist. And she's a lot older than me. She was already a full professor at UC San Diego at the time. She was in the UK on sabbatical. Anyway, so through her, first of all, I kind of accidentally learned a lot of biology over the dinner table, the way you do. But also, after a couple of years, I began to realize that we were just never talking about aging. And I realized that this was very strange because I had always known that even if the problem of work is a big problem, the problem of aging is a much, much bigger problem. And I had always assumed that people who were talented at biology would be working on it because they would think the same. It turned out that no, she wasn't really interested in aging. And it wasn't just her, it was true of all the other biologists I met through her. And so it took me a couple of years to really, get my head around this crazy fact that people didn't really appreciate that aging was actually quite a bad problem and furthermore a problem that could potentially be addressed. But eventually I decided, well, okay, I've got no choice. I've got to switch fields. And by this time, first of all, I had done quite well in my AI research, so I knew that I was good at working on really hard problems. And secondly, I knew that there was quite a distinguished history of scientists working and becoming established in one field and then switching fields and doing really important work in their new field as a result of perhaps just kind of not being encumbered by the conventional wisdom of the new field. Um, so the best example, the best known example is that the whole of molecular biology was invented in the 1950s by a bunch of physicists. Um, yeah, so that's basically, I decided, I've got a chance of making a contribution. So, and I happened to be in a position at that point where I could just switch fields without really taking any risk, because I had a nice, well-paid and very undemanding bioinformatics job that gave me a lot of spare time and access to university facilities and everything. So that's what I did in the mid-90s, and kind of the rest is history, I guess.
And this was at Cambridge, right?
That's at Cambridge. That's right. Yes. So I got my undergrad degree in Cambridge in 1985, and eventually I also got my PhD in biology from Cambridge in I think 2000 on account of basically submitting the work from my first half dozen papers or so.
So how has your background in computer scientist and in automation and artificial intelligence contributed to your approach toward longevity?
So I would say that it doesn't really make much of a contribution these days, but in the early days,
Mhm.
shortly after I switched field, it made a huge difference, and exactly the same kind of difference that I mentioned earlier about switching fields. I came at the problem of developing medicine to actually address aging from a completely different angle than the people who were working in the field. And in particular, that's really what led after a few years to what I think I'm mostly known for in the field, which is pioneering the idea that repairing the molecular and cellular damage of aging is actually going to be easier than making the body run more cleanly and slowing down the rate at which the body generates that damage in the first place, which is where everybody else had been focused. That was really very counterintuitive to the field, and it took me best part of a decade, I would say, to really get my colleagues to understand that this made sense. It's completely mainstream now, but it was a big paradigm shift.
What other paradigm shifts have you noticed over the last 20 years in the industry?
Well, in terms of conceptual shifts,
Mm-hmm.
there's really nothing that compares with that one. That was really big. But in terms of attitudes, generally the acceptability of thinking about aging as a medical problem that has the potential to be addressed very comprehensively, that's been a big paradigm shift, in people's attitudes over, especially the past 10 years. And it's been manifest in the, of course, academic community and the research community, but increasingly it's been manifest in the wider world. First of all, I guess a big thing happened around 6 or 8 years ago when investors started getting involved. People who actually were interested not only in furthering the research, but actually making money out of it in the foreseeable future. You only get to that point when things are looking reasonably close, because of course anyone who wants to make money, they want to make it tomorrow. Um, so that was a huge thing. And as time's gone on, especially the IT community, the people who come from the same kind of background that I do, have increasingly got on board. They're kind of like the paradigm of damage repair and preventative maintenance, if you like, is something that sounds very straightforward and commonsensical to geeks. And so more and more of them have been coming on board. And of course, these days we have the great advantage that there are quite a lot of geeks out there who are both wealthy and young, and so have not lost the ability to aim high, so to speak. And that has certainly been transformative as well.
Talking about young technology talent diving into the longevity industry and the recent developments in artificial intelligence, what do you think are the roles of artificial intelligence in the longevity industry? Is this just buzz? Are there any real applications? Is this just media coverage? What's your take on that?
Yeah, so the role of AI in the longevity movement is enormous. But as with a lot of things that are not just AI, The most important ones are not actually specific to longevity. They apply across the whole of biomedical research. So in the same way that, for example, the development of induced pluripotent stem cells 15 years ago, or the development of CRISPR 10 years ago, these apply across the board. They're certainly enormously relevant to, to excuse me, to longevity research, but they're also equally relevant across all medical research. So similarly, a couple of years ago, of course, there was this massive breakthrough by DeepMind developing a program that was able to more or less solve this very long-standing thing called the protein folding problem, the figuring out of the three-dimensional structure of a protein just from its amino acid sequence. And again, this is something that applies, it will enormously speed up pretty much all medical research, but certainly it will absolutely have an enormous impact on longevity research. Outside of that, there are plenty of other things. So, for example, my great friend Alex Zavaronkov has a company in silico medicine, which is one of the biggest companies in this space now. And their main focus is using state-of-the-art machine learning techniques to identify drugs against aging. And, of course, not just new drugs, but also to repurpose existing drugs. There are various other companies doing that kind of thing. And again, their technologies can be applied across other areas of biomedical research, but Alex and his company are mainly focused on aging.
Yeah, that's a very promising company. They recently had a deal with Sanofi.
Yep.
That entitles them to up to $1.2 billion in milestone payments.
Yep.
It's a really interesting field. Now, why don't we dive into the damage repair approach that you pioneered? Can you please explain this approach? I understand you have a classification of age-related damages that you famously call the 7 deadly things. Can we dive into that and go
Sure,
over
yes, absolutely.
damages?
So the best thing to do by way of getting going in that is to explain why it was so difficult for my colleagues in the gerontology field to understand really what I was saying. So if you ask the question, what is aging? First of all, you'll get a ridiculously large number of different answers, even from experts. But a perfectly good and uncontroversial definition of aging, which I use all the time, is that it consists of two processes. One process that goes on throughout life, and another process that kicks off late in life, in middle age or later. So the first process, the lifelong one, is the creation of damage in the body by metabolism. So our metabolism is simply the word that biologists use, of course, to describe the entire network of processes that keep us alive from one day to the next. And damage is simply molecular and cellular changes to the structure and composition of the body that are brought about as consequences of metabolic processes. Now, the reason it's appropriate to use the word damage to describe those changes is because they are progressive. They accumulate over time, and the body is only set up to tolerate a certain amount of those changes, which means that eventually that threshold is exceeded, and that's when the second process kicks off, where the damage starts to impair the function of the body, whether physiological or cognitive. And so that's the second process. So the first process is metabolism creating damage, happens throughout life. Second process is damage creates pathology, happens late in life. All right, so the body's really, really complicated. Metabolism is ridiculously complicated, so no surprise, there are lots of different types of damage. And of course, there are lots and lots and lots of different types of pathology of late life. Now, the whole field of gerontology really got going more than a century ago when people started to realize, a few people, that you couldn't just go after the pathologies of late life in the same way that you would go after infections. They were just too numerous, and they would happen at more or less the same time. They would interact with each other and exacerbate each other. It was just too big a problem, too many different things that you'd have to address all at the same time. And so the concept of gerontology was to go back in the chain of events, to to try to find some kind of unified cause of all of these things and attack the thing at this early stage in the chain of events. But essentially what gerontology did, it went too far in that direction. tried to go back and try to, if you like, manipulate metabolism so as to somehow make it stop doing the thing that we don't want it to do, the creation of damage, while at the same time still letting it do the things we need it to do to keep us alive. And it turned out that that just wasn't possible, that these two things are just too inextricably intertwined, and it's just like, it's a non-starter. So by the 1960s or 70s, it had become completely unacceptable to even think and talk in a grant application, for example, about doing anything about aging. Gerontologists had become a bit like seismologists, where they know the thing they work on is bad for you, but they don't really have any aspiration to actually fixing it, stopping it from happening. Yeah, so then when I came along, I said, well, okay, wait a minute, let's just attack the damage, this thing in the middle, right? People who were fixated on trying to mess with metabolism, even though they had basically decided that it was a non-starter because it was too difficult, they still looked at it and said, well, okay, hang on, isn't this just geriatrics again? Isn't this just trying to go after things one at a time because there are lots of different types of damage? And it took a long time for me to get people to understand that, no, it's not the same. Because the thing about the pathologies of late life, it is not only that there's so many of them, it's also that they are the consequences of something that's accumulating, namely damage, right? Which means that, by definition, even if you're attacking all of them, even if you've got all of these things, nevertheless, anything that you do that attacks the consequence of something that's accumulating is inherently going to become progressively less effective as the person gets older. So it was really a non-starter. Plus also you have the enormous advantage that damage, while it's accumulating before it's reached this threshold that's pathogenic, damage is basically inert. So different types of damage don't interact with each other, or at least very little, whereas pathologies interact a hell of a lot with each other. So again, the problem becomes vastly simpler. So that's the main thing that allowed me to do this. But then, of course, I've still got to go a bit further. So now to answer the question you asked about the 7 deadly things. But even though damage is a lot more easy to address than pathology, nevertheless, you've still got lots of them, and you've got to develop ways to fix them. And really, that only becomes practical because it's possible to classify these many types of damage into a manageable number of categories. So as you say, I talk about 7 different categories. So what is the purpose of this classification? The purpose is to identify and highlight repair approaches. Remember, the idea here is not simply to slow down the rate at which damage is created, but actually to eliminate that damage every so often so that it doesn't reach the pathogenic threshold. And so for each of these categories, even though there may be many examples within a category, there will be a generic approach to repairing that type of damage that can be applied with differences only in details between the different types of repair. So a simple example would be cell loss, where cells are dying and not being automatically replaced by the division of other cells, right? So, of course, the standard repair strategy for that is simply stem cell therapy. So if we take the example of Parkinson's disease, which is an aspect of aging, a pathology of aging, which is mostly driven by that one type of damage, the loss of dopaminergic neurons, people are now in clinical trials in Japan and a couple more starting up in the US to do exactly this, to put in stem cells, dopaminergic precursors that will differentiate to replace the cells that the body is not replacing on its own and thereby restore dopamine and get rid of Parkinson's. And the same kind of thing can be done in, for example, the thymus, which atrophies relatively early in life with the eventual result that we don't have enough naive T cells. So there are other ways that may work just by making the residual cells in the thymus more stem-like and getting them to regenerate the thymus in situ without stem cells. But the same thing generally applied. You're increasing the number of regenerative cells. And the same kind of thing applies across the other 6 types of categories of damage. There may be many examples of which ones, and you may have to develop different therapies, but the therapies will have a lot in common, which means that once you get 1 or 2 of them working, then it's going to be that much easier and faster to get the next one working and the one after that, simply because you're going to be able to reuse the expertise, the knowledge that you gained in the first 1 or 2.
So using the damage repair approach as a framework, is it fair to compare the the aging body to an aging machine or an aging car, and something that needs periodic maintenance?
I believe it's extremely fair, yes. People often look at the analogy between the living organisms and simple man-made machines, and they say, no, this is a very oversimplistic analogy, and you really can't stretch it too far. But they're wrong. I really believe they're wrong, that the analogy is extremely strong. The only real differences, the only real things that make the analogy look as though it's not so strong number one, that the human body is vastly, vastly more complicated than any man-made machine. Plus, of course, we don't have the blueprint, right? So it's not so easy to. We have to kind of reverse engineer things. Um, um, but that's the difference of degree, right? That's the reason why we can already keep cars going for 100 years, even when they were designed to last only 10 years, whereas we can't do that for a human body yet. It's the difference of degree. That's all. The other difference that people point to is they say, well, but the human body's got all of this enormous arsenal of automatic, inbuilt self-repair machinery, and machines don't have that. And that's kind of looking at it backwards, because actually the fact that we have this inbuilt self-repair machinery makes it easier for us to do what we want to do, because all we're doing with this regenerative medicine, with this preventative medicine, damage repair medicine, is to augment that machinery to essentially remove the gap, to fill in the gaps, to remove the types of damage that the body does not have ability to remove on its own.
No, that's an excellent point. We can develop therapies that take advantage of the endogenous repair processes of the body.
Absolutely, we're on the same side here, that's right. So there are cases, for example, where waste products will accumulate in the body, and we just want to get rid of them. We may only have to make one chemical step, one enzymatic step to to convert this such a molecule into something that the body already has the ability to break down or to excrete. We don't have to like mineralize the whole thing with a whole bunch of different enzymes. We can just make one step.
Okay, so Aubrey, you pioneered this approach in the late 90s and early 2000s at the turn of the century. And then in 2013, a seminal paper was published called The Hallmarks of Aging, that it has thousands of citations, and that also organized the processes that contribute to aging in 9 categories, and now it was recently expanded into 12 categories. So how does these biological processes considered in the hallmarks of aging compare to the damage approach? Is there a direct relationship, or is it a more complex relationship?
Yeah, there's a very close relationship. So as you say, this paper that came out in 2013 is far and away the most highly cited paper in the whole of the biology of aging this century. And there's been, as you mentioned, a recent update of it by the same authors. There's also been a whole bunch of other papers saying more or less the same thing, calling them pillars of aging or expanding to like 14 hallmarks or whatever. The comparison with the work that I did a decade earlier is pretty tight, actually. Essentially, most of the stuff that's talked about in these papers is damage repair. They talk about, well, it's damage and corresponding damage repair. So that's great, and certainly it has meant that since 2013, I basically never need to justify the damage repair paradigm to within the field, whereas I had to justify it quite a lot in the previous decade. So that's all great. However, there is one really big shortcoming of the Hallmarks paper and really all of its copycat papers that happened over the subsequent decade, which is that it doesn't really focus on this correspondence that I talked about a moment ago between category of damage and a type of repair. There's plenty of kind of allusion to that kind of thing, but the overall structure of the classification does not have that at its foundation. And that's quite a bad difference, because at the end of the day, you want this classification to be useful. You want it to actually guide some kind of, to provide some kind of guide to what medical researchers need to be highly prioritized. And one thing that did lead to, which has not really been properly eliminated even now, is some omissions from the hallmarks paper, some types of damage that have not really been appropriately highlighted. So one thing that particularly is conspicuous here is the accumulation of waste products, molecular waste products. In the original hallmarks paper, there was a very important discussion of that, but it was restricted to waste products that are made of protein. So I think the official name of the category in the hallmarks paper was loss of proteostasis. And certainly that's very important. But there are other waste products that are not proteinaceous and which also cause problems. There is, for example, a derivative of vitamin A that accumulates in the retina and is the main driver of macular degeneration, the number one cause of blindness in the elderly. Similarly, there are oxidized versions of cholesterol, which are poisonous to white blood cells, to macrophages, and which are the main driver of atherosclerosis, obviously the number one killer in the Western world. These things were really not addressed at all, and honestly they're not really addressed even now in the more recent papers. Another example is the loss of elasticity of the extracellular matrix. That was completely left out of the first hallmarks paper. It has been kind of introduced, but I think it could have been introduced a little bit more explicitly in the more recent versions of this. So yeah, basically, of course, I'm a bit biased, but at the same time, yeah, I do think that my original classification, which was published in 2002, is still a better one than the ones that have been coming out more recently.
Another point that you mentioned earlier is that the damage types do not interact with each other because they are more fundamental, but the pathologies do. In contrast, the hallmarks of aging are completely interconnected. Even the authors say that all the hallmarks have a relationship within each other. Is there any particular hallmark of aging that, in your opinion, is a more attractive therapeutic target or would have a more significant impact in health? For example, a lot of people have commented on proteostasis because it's a fundamental process. Other people say to focus on genes. What's your take on that, if any?
Yeah, so I prefer not to try to prioritize the different types of damage, different categories of damage in terms of their importance, because when I say that we don't have to worry so much about the interactions between types of damage as we would in terms of the interactions between pathologies, I don't want to take that too far. There is some interaction. And in fact, in some cases, the interaction may be quite substantial. So I think everyone was really taken by surprise when the first studies started to come out looking at the elimination of senescent cells in mice. As you probably remember, there was this brilliant collaboration between the group of Jim Kirkland and Jan van Dersen at the Mayo Clinic and Judy Campisi at the Buck Institute. Came out in 2011, I think. In which they created a genetic model in which senescent cells would accumulate at an unusually rapid rate, and they could eliminate those cells just by giving the mice tamoxifen or something like that. The salutary effects, the beneficial effects on these mice from removing the senescent cells were much more widespread than anyone had really expected. So this was an enormous validation of the idea that Judy had pioneered in the late 90s, that senescent cells might be actively toxic rather than simply being useless. In other words, the concept of this thing called the SASP, the senescence-associated secretory phenotype. And it shows that even relatively early in life, before senescent cells or before the pathologies of anything have started to emerge, there may be some substantial interactions between the types of damage. Now, my guess is that most types of damage do not have that pervasive an interaction. Mitochondrial mutations might have, but we aren't there yet to know really whether that's true. However, the main thing is that we still probably don't need to worry too much about these interactions when we are designing the damage repair interventions. We should be able to identify and optimize the damage repairs of each of these things as relatively independent things, and then combine those things when we have them reasonably working. And that, of course, is the focus of the flagship project of my new foundation.
Well, let's talk about your new foundation and combination therapies. It's Longevity Escape Velocity Foundation. Can we start by explaining to the audience what's longevity escape velocity? What's that
Yeah,
concept?
sure. So this is one of the terms that I coined a long time ago, nearly 20 years ago now. Um, Essentially, it comes down to the consequences for healthy longevity and indeed total longevity that arise from looking at damage repair therapies rather than um, damage retardation therapies, the sort of thing that came before I introduced the damage repair concept. Um, Essentially, the thing about damage repair is it buys time. If you take someone who is, let's say, 60 years old, then if you have a reasonably comprehensive damage repair panel of therapies, but it's not perfect, then you may be able to give these therapies to this 60-year-old, and they won't be biologically 60 again until they're, let's say, 80, right? Now, what do I mean by biologically 60 again? What I mean is having the same amount of damage in the body as they had when they were 60. Now, of course, I'm oversimplifying there to some extent because the types of damage will be, a bit different. And that's actually the point. The, the fact that the panel of therapies is not perfect, what that means is that there's a bunch of damage that the therapies repair, let's call that the easy damage, and then there's a bunch of damage that the therapies don't work on, the difficult damage. So when someone gets to 80, and they are biologically 60 again, it's not because they got the therapies once and they need to have the same therapies again. They could have the therapies every month. And so basically that means that there's essentially zero easy damage in this person's body ever from the age of 60. But the difficult damage is not being repaired, so it's continuing to accumulate. So by the time they hit 80, they've got a 60-year-old's amount of damage, but it's all difficult, which means that however often you carry on giving these therapies, they're not going to help. The person's going to carry on getting more and more damaged and sicker and so on. But the thing is, we've had those 20 years for people like myself to improve the therapies, and that's going to work. We're going to make progress during those 20 years, which means that actually the 80-year-old who's biologically 60 for the second time is going to be amenable to re-rejuvenation. That we're going to be able to give them therapies that repair not only the easy damage, but also some of the difficult damage, so that we can take them back again, so that they won't need a third rejuvenation or further improved therapies until they're, let's say, 100 or more. So that means that we can define this concept of longevity escape velocity, which is simply the minimum rate at which the scientific community needs to improve the comprehensiveness of these damage repair therapies so as to stay one step ahead of the problem, so that people who are always receiving the latest therapies will indefinitely have a level of damage in their body that is below the threshold and does not trigger any of the pathologies of late life. And the great news is that rate that we need to improve the therapies slows down over time. There's really no concept of that rate at this point. You have to actually get to longevity escape velocity first, and that's a threshold that I believe we have a 50-50 chance of reaching within the next 15 years or so. However, once we're there, because things get easier thereafter, there is essentially zero chance that we will ever fall back below longevity escape velocity once we reach it. So a friend of mine many years ago coined the term the Methuselarity by analogy with the singularity. They said, there'll be this point where we reach this point where aging is basically over, and it's functionally as if we had completely 100% perfect damage repair, even though we don't.
Oh, that sounds exciting. So you can probably quantify longevity scale velocity on biological age, correct? You need very precise metrics of biological age to keep track of your damage, or
Yeah,
how would
not
you
really.
say that you
Yeah,
when can you say that you reached the longevity escape velocity?
so this is a good question, but it's actually a question that I don't focus on very much.
Hmm.
Um, the problem with measuring biological age is that some things are a lot easier to measure than others. And so we end up with these proxies, these things that do change with time, with chronological age, and they change pretty uniformly. Of course, there's a great deal of interest these days in epigenetic clocks, which have been developed over the past few years. Um, but there's plenty of other measures, including simply functional ones like grip strength and gait speed and so on. And all of these things have, um, you know, a good correlation with chronological age, and sure enough, also with biological age as measured, like predictively. In other words, as an indicator of how long you're likely to live, how long you're likely to stay healthy and not exhibit the health problems of late life, and so on. But they're all proxies, or some of them are more proxy than others, more indirect than others. Now, I'm fine with that. I think that we certainly need to identify all of the various types of damage, all of the molecular and cellular changes that we need to address. And obviously, whenever we develop a therapy that's supposed to eliminate a type of damage, we'd really like to be able to measure how well it's working as directly as possible. But sometimes it's just not going to be direct. And, we have to kind of live with that and just, take it somewhat on faith that we really are eliminating the type of damage that we want to eliminate. And of course, as time goes on, we can get longitudinal data on people who are, receiving these things and see how well we are postponing the health problems of their late life. But in a sense, it's correct to say that we will not really know at once when we reach the Methuselarity. But that's okay.
So what steps is your new foundation taking to reach this longevity escape velocity? What is your mission?
Yeah, so it's a little paradoxical. So LEV Foundation is a non-profit focused on early stage research. So we do experiments in mice, and we'll probably do experiments in cell culture as well, same as SENS Research Foundation does. But we certainly don't do clinical trials. And the reason why this is a bit paradoxical is because we will never reach longevity escape velocity for mice. Essentially, shorter lived organisms, you would need to improve the comprehensiveness of the therapies impractically rapidly. It's touch and go whether we will ever reach longevity escape velocity for cats and dogs. Certainly not for mice. And yet mice are the things we experiment on, which is a little odd, I guess. But yes, what we're doing, of course, is we are developing therapies that get as far as one can towards longevity escape velocity for mice. In other words, that postpone the health problems of their late life. And in particular, we are doing this by damage repair therapies that are applied when the mice are already in middle age. So our goal here is, and of course we're combining different therapies that address different types of damage. So our goal here is to reach another milestone that I define called robust mouse rejuvenation. And these days the way I define that is I say we have to take, first of all, a nice long-lived strain that lives at least 2.5 years on average and lives maybe 3.25 years maximum. And then we have to take those mice when they've got about a year left to live, so typically when they're on average about 18, when they're 18 months old, and we want to add a year to that, both to the mean and to the maximum. If we do that, then we will unequivocally have had a dramatic impact on the aging of these mice. If you take mice today, and you do anything that's ever been done to mice at 18 months of age, then we can achieve maybe a quarter or a third of that amount of increase. And so we want to do a lot better than what's been achieved so far. And I believe that there are two reasons why that's important. Number one, it will genuinely, as with all mouse work in medical research, it will genuinely give information about what's likely to work in humans. Now, of course, we know that that information will be very tentative. We know that the hit rate of translatability from mice to humans in medical research is pretty low. Realistically, I think it is fair to be somewhat optimistic and to say that damage repair approaches will translate a little bit more um, confidently to across species than drugs do, because than things that mess with metabolism, so to speak, because damage is more similar. Um, but the other thing is rhetorical, of course. The other benefit of this, if if we're out there being able to do this much to double the remaining lifespan of middle-aged mice, then it really becomes completely impossible for the naysayers to go out there and say, yeah, aging is just not, it's not appropriate to think of it as a medical problem. It's like gravity, it's inevitable, it's woven into the fabric of the universe, and just, stop working on it. People just won't be able to do that anymore. And that will be huge in terms of the ability of especially policymakers to take the public with them, to see that there are votes involved in actually putting serious money into the medical research, and not just the medical research, but the infrastructure, the building, training of medical personnel, and so on, so that once these therapies do actually become ready for prime time, that they can be disseminated as rapidly as possible to everybody who's old enough to need them.
So what kind of therapies are you focusing on at the LEVF Foundation? You mentioned combination therapies.
That's right. So at SENS Research Foundation, our focus was always to work on individual types of damage, and especially we would prioritize the ones that were being neglected by other people, the ones that tended to be most difficult to address, and therefore, least amenable to making money out of them, or for that matter, even getting high-profile publications from, positive results. So, of course, SENS Research Foundation was set up as a charity overwhelmingly funded by philanthropy rather than by grants or investments. So at LEV Foundation, we have a somewhat different focus because we feel that that problem, the damage repair rejuvenation challenge, has pretty much been solved. That pretty much everything within the development of damage repair therapies for particular types of damage has now progressed sufficiently far that other people are working on it a lot. Even the most difficult things are now leading to actual startup companies that are reasonably well funded. So, there's still stuff to do, and that's why SENS Research Foundation's work is still very valuable. But there's not all that much to do. It's really pretty much mainstream now, and I don't do mainstream. My strength is being able to go out there and be the professional heretic, and take all the bullets and open the doors for people to walk through. And so at LEV Foundation, we are very much focused on combining therapies. This is something that even today is still very orthogonal to the incentive structures of either of industry or of academia. In industry, combining different things where some of them may not even be patentable, some of them, even if they are, the IP is owned by different companies, there's all manner of complicated cross-licensing and such like. It's just like it's ugly, right? And then from the point of view of academia, again, you don't get papers in Science or Nature or Cell if you just combine things that already work somewhat and they work better. It's just not exciting. It's not hypothesis-driven. It's kind of, again, doesn't fit the incentive structure. So we are, filling that vacuum by taking things that already work, where other groups have already shown substantial improvements individually with therapies applied in middle age, and the benefit is shown in terms of both mean and maximum lifespan in mice. And we're putting those things together. We're starting out in round one of this research program with 4 different therapies, very different ones, that are attacking different types of aging, different aspects of aging. And we have no idea what's going to happen, but we're combining them in various combinations, and we're hoping that we will get a very substantial result. However, our goal is to do a lot of these things. If this experiment even slightly works, then the first round won't be done for 2 years. So we definitely want, in less than 1 year, to start round 2 because there's plenty of other candidates out there, things that we haven't had the opportunity to include in round one, and that again we want to combine and get more information. And of course, we'll be folding in information as it comes out from the community all the time as well.
Can we please talk briefly about these four therapies in round one? Are they off-patent drugs?
So let me go through the things that are included in round one. The first one is not actually a damage repair therapy. We're putting it in as a kind of non-repair control, if you like, and that's rapamycin. So of course, rapamycin is a very high-profile drug that's been around for a long time, but recently relatively recently, was established to be a really quite powerful calorie restriction mimetic, a drug that essentially tricks the body into thinking it's on in a famine when it isn't. And the amount of life extension that was obtained in, honestly, an experiment that was done by accident by the NIA, starting at 20 months of age in mice, was comparable to what you would get by bona fide calorie restriction in mice starting at that same age. And this is the first time that that had been achieved. Other drugs that had this capacity to mimic calorie restriction would only mimic it to a much lesser extent. So this was very exciting, and people have been desperately going after analogues of rapamycin that they can actually get patents on and so on. And it's all very exciting. So that's one thing we're including. The second one we're including is a kind of variation on the theme of heterochronic parabiosis. So this is another thing that's been, of course, very high profile for the past 15 years or more, where if you connect the circulatory systems of a young mouse and an old mouse, then the old mouse is rejuvenated in many different ways. Since that's not therapeutically practical, people have been interested in going further, and there's a lot of work been done recently on plasma exchange. We funded some of the pioneering work in that area at SENS Research Foundation in the Convoy Lab. And so, basically taking out old plasma, putting in new plasma. Now people have moved on to taking out old plasma and putting in just basically saline with albumin in it, and that's also quite beneficial. But it turns out that the thing, the variation on this that has had the most impact on actual mean and maximum lifespan in mice so far is not messing with the plasma, but instead messing with the cells in the blood. What was done was to do a bone marrow transplant heterochronically, in other words, mobilize the bone marrow cells in an old animal and put in a whole bunch of bone marrow cells taken from a young animal and allow those ones to re-engraft. So of course some of the old cells that were mobilized would re-engraft as well, but you get plenty of the young cells engrafting, and this achieved quite a substantial life extension a couple of years ago now. So we are including that as one of our therapies. Next one is telomerase, and this is a really curious one because people for the longest time said to themselves, well, you know what, if you overexpress telomerase in mice, that's not going to make any difference because mice, unlike humans, they make a lot of telomerase. Most of their cell types express it promiscuously. So, and the telomeres are really long, so what's the point? But of course, sometimes people are not actually right when they think, they sometimes oversimplify. And sure enough, several years ago now, my great friend Maria Blasco from Spain, who was one of the pioneers of working on manipulation of telomerase in mice, did the right experiment. She did an actual gene therapy experiment with telomerase in mice. And what do you know? It did actually have a beneficial effect. Took a while to figure out really what was going on, and indeed Maria was the person who really nailed this. She was able to establish that the rate at which telomeres shorten in mice, just per cell division, is much, much faster than it is in longer-lived organisms like humans. So basically, telomeres are just very unstable in mice, and you need a bunch of telomerase just to hold things together. So we kind of understand what's going on there now. But anyway, the point is that again, she was able to get life extension, and much more recently, Another group, George Church and Liz Parrish led it. They did the same kind of thing using a different delivery mechanism, a different vector. And again, they got very good life extension. So that's another one that we're including. And the final one is senolytic. So as I mentioned earlier, people have been very excited for more than a decade now at how wide-ranging are the benefits from removing senescent cells in mice. And people have been desperately trying to figure out, trying to identify drugs and supplements that that selectively knock out senescent cells. Honestly, there's been a big problem of reproducibility in the senolytic field, and there's a long way to go really before we have such things. But another Spaniard who actually used to be married to Maria Blasco, Manuel Serrano, had a brilliant idea several years ago. So one thing that's known about senescent cells, this is not a universal marker, but it's a pretty good marker, is that they have a very high expression of this one enzyme called senescence-associated beta-galactosidase. And what does that do? It breaks down a sugar molecule called galactose. Right. So Manuel thought, I know, what we'll do is we'll take a senolytic that's basically just bad for cells in general, right? But maybe it's, slightly worse for senescent cells, but, he recognized the reproducibility problem and the therapeutic index problem. So he said, well, let's make it more selective to senescent cells by encasing it in galactose. Just so that when it goes into non-senescent cells, it won't do anything because it won't get out of the cage that it's in. Whereas in senescent cells, the beta-galactosidase will break down the galactose and the molecule will be liberated and it'll kill the cell. And he did this with a senolytic that had been already slightly well studied called navitoclax. And it pretty much worked. It worked. It seemed pretty effective. More recently, that work has moved on to a second generation version of it, which is much more reproducible. And here, what happens is, rather than encasing the drug in galactose, instead we use an actual covalent conjugation. So we make it into a prodrug. Just one molecule of galactose is attached to a particular point in this thing, navitoclax, and it's attached in a place which basically inactivates the navitoclax. But again, beta-galactosidase can come in and cleave off and break down the galactose, and the drug is activated, and bang, your cell dies. So we are using that conjugated navitoclax as our senolytic. Those are the four things.
So are you generating new IP? Are these new senolytic, for example? Do you plan to license it? How would that work with the foundation?
Right. So LEV Foundation is a non-profit. We're a 501c3.
Mm-hmm.
And all of the things that I've just spoken about have been published by other groups. Oh. So, for example, this conjugated navitoclax is absolutely, it's been patented by somebody else. Oh. Right? So we are not looking at IP at this point, not at LEV Foundation. At SENS Research Foundation, as soon as the private sector started really getting involved, we basically pivoted to a business model in which we would be rather aggressive about IP, about obtaining IP, so that we would be in a position to spin projects out as startup companies. And we did that half a dozen times. It was very effective. But I don't see us doing that at LEV Foundation in the near future anyway.
Got it. Thank you for clarifying. So talking about doing the right experiments the right way and the reproducibility crisis, which affects every discipline in science. But since longevity science is doing generally very cutting edge stuff, do you think it particularly affects longevity science? Would this damages the credibility of any field?
So, I mean, there is a huge problem across the whole of science, as you say, with the way that it's funded. Honestly, we hear about these occasional cases of bona fide fraud. It's absolutely astonishing that fraud is so rare in science, because the pressure to manipulate one's data is just astronomical. With the huge shortfall of money, the desperation that people have to actually get positive, high-profile results in order to get their next grant application funded. It's really an enormous testament to the integrity of the scientific community that there is so little fraud. But, of course, if we go back and we talk about kind of something much less than fraud, just kind of, highlighting particular things in one's results and downplaying others, then this is enormous. And the the publications, the publishers have an enormous role to play here as well, because the biggest thing is that the high-profile journals are very, very reluctant to publish negative results saying, we tried this, it didn't work. The whole term negative result is kind of it should be banned, because there's no such thing. You learn shit, right? But it makes it very, very difficult, and this hugely biases what experiments people do in the first place. They will do experiments that are low-hanging fruit, that are likely to get a positive result very quickly, even if the researchers know perfectly well that it's not really the right experiment to do because it won't scale, it won't lead to the next good experiment to do, it won't actually be the right thing to do. Honestly, I'm not saying that I really blame any individual sector of the community, any individual constituency, because it's really the classical prisoner's dilemma thing. The public doesn't understand science, so there's not enough public money because the governments want to get re-elected, and then because there's not enough money, you've got the academics having to, as I say, focus on low-hanging fruit, and when they go out and talk to the media, they have to sometimes overhype, if you like, the significance of their results. Most scientists don't get on TV every week the way I do. They get they get on TV like twice in their career, and they've got to do their best. So I have sympathy with everybody who's involved, but still, it's pretty bad. And as you say, this does lead to problems of reproducibility as well. That people will do an experiment in a way that maximizes their chance of getting a positive result, and sometimes that means cutting corners on like control experiments and so on. So yeah, it's tough.
So what steps can be taken to address this crisis? Is it a matter of funding, or is it just the whole structure of publications, government grants? How can we tackle this?
It's mostly funding, especially public funding, which is where most funding for academia for the early stage research comes from. Essentially, in the old days, in the 18th century, when scientists were funded by patrons, you didn't really have this problem in the same way. People could just work for as long as it took to get where they were going. And if they didn't get anywhere, well, at least they tried. And that's what science is about. It's not about, being the person who gets the glory. It's about being part of the army that gets, and someone gets there. So yeah, I think having a lot more money and a lot more reliable money, as opposed to short-term grants. In fact, I wrote something several years ago about this that basically said that people should not be given grant money on the basis of what they propose to do with it, but rather on the basis of what they've done in the past. That, and especially what they've done in the relatively distant past. Of course, this has to be somehow seeded, so a beginning scientist has to be given some kind of initial pot of money to get going. But once you've done that, you should be judged on your record much more than on what you want to do, because what you want to do, you're the expert, right? Otherwise, it wouldn't be novel, right? And so convincing a bunch of your colleagues that what you want to do is worthy of funding. That's a mug's game. That's an uphill battle, and they're just gonna not understand everything. They're just gonna be wrong sometimes, especially in the most important work, which tends to be cross-disciplinary. To bring ideas together, which nobody has ever brought together before. So none of the experts who are evaluating your grant will know all of the facts that they need to know in order to make a good evaluation. So yeah, I just don't believe in grants funding being allocated on the basis of saying what you're going to do with the money.
So is there a role for private investors, like venture capital and private equity, high net worth individuals to fund these longevity biotechnology companies? Because in the end, the idea is to translate the research into useful therapies, right?
Yeah. So It's a bit of a dilemma because, on the one hand, so I mentioned earlier, perhaps a little bit flippantly, that anyone who wants to make money wants to make it tomorrow. So there's an inherent short-termism in there. Of course, that's not 100% true. So if we look at Calico, for example, Larry Page and Sergey Brin say they put a lot of money into aging, and they kind of said, we're going to take a long-term view. We don't care how long it takes. They almost went too far with that and kind of, treated it as a kind of badge of honor that they didn't have a short-termist focus. And the result is basically they don't have, they more or less have a valley of death within the company, that they don't really have a pipeline for the timely translation of scientific knowledge into the clinic. Um, and now with Altos, we're probably seeing the same kind of thing. Jeff Bezos has been interested in aging for at least 15 years since I met him at TED, and he has now finally decided to put his toe in the water. But again, he's done it in a private company, but it's probably not going to really work like a typical private company. It's going to have this same long-term focus like Calico. so, so in that sense, the private sector can certainly help. In other senses, when we look at the more general investor market, where we do have an inherent, structural requirement to make some money soon, I think the best way to look at it is in terms of early exits. In other words, not looking at a startup company and thinking in terms of the eventual profitability of products that will come out, but rather thinking in terms of the point at which big pharma will acquire the company, or the point at which the company can go IPO, and one can one can have profits that way. These things certainly, as the industry becomes more mature, are going to burgeon. Big pharma is still at the very beginning of taking proper notice of the longevity world. And, so there's a couple of cases, just really hardly any cases so far of acquisitions, but that's going to snowball over the next couple of years. And so if investors of every type who are interested in startups are thinking in those terms, then it's going to be a much more attractive proposition.
Got it. So it's there are two games to play: the short term game with early exits, and then there's the long term game with more perhaps radical stuff. In terms of radical technologies and radical treatments, are you particularly excited about any biotech companies in general? We mentioned Altus.
I'm extremely excited about a lot of biotech companies, because the damage repair approach is a divide and conquer approach. I'm constantly keeping my finger on a lot of pulses. But yeah, some of them are particularly exciting. One that I always like to mention is Maya Technology, which is in Chicago, and they have a number of anti-cancer approaches. But the one that excites me the most is called THIO, THIO. It is a drug that essentially kills cells that are expressing high levels of telomerase. So this, of course, 90% of cancers do this. People are very fond of saying, cancer is not just one disease. It's, it's a whole bunch of different diseases with different gene expression patterns and so on. But of course, that doesn't contradict the fact that cancers do have some things in common, and in particular, 90% of human cancers have this one thing, this very high expression of this one enzyme. So this drug basically causes cells that are making this enzyme to kill themselves rapidly. And it appears to have a very nice therapeutic index because the non-cancer cells that express telomerase in the body, such as, for example, the long-term repopulating cells in the blood, they express teeny tiny trace levels. So if you're just a little bit careful with dosage and duration of therapy, then everything seems to be good. So they're recruiting for phase 2 right now, this company, and I think it's the single most exciting thing in the whole of cancer research right now. There's a company called Nanotics that I really like, which is focused on a really important and hitherto pretty much inaccessible aspect of medicine, which is to be able to distinguish between two versions of the same protein. One version that is bound to the surface of a cell, and the other version that is circulating freely in the bloodstream. In many cases, you want to be able to eliminate the stuff that's circulating freely without inhibiting or harming the one that's bound to the cell surface. And Nanotics has a brilliant technology that's very generic for doing this. They're using it for cancer, for sepsis, for a bunch of other things. They've got collaborations with the Cleveland Clinic and the Mayo Clinic and other groups. So they're doing very well. But the faster they grow and the faster they can do more, the happier I will be. It's wonderful technology.
Yeah, Nanotics is actually one of quadraScope's portfolio companies.
Great.
And,
Great.
I had the opportunity to interview the CEO, Louis Hawthorne.
Mm-hmm.
So you are clearly a public figure, and you go to many events to create awareness about the longevity industry. Can you give us some recommendations of some events to attend, perhaps for a lay audience, perhaps for scientists?
So, yeah, one of the great things about the burgeoning of this industry over the past several years is that there are now a lot of conferences out there that are really, really vibrant and worth going to. I have, of course, resumed running my own conferences, which came to a juddering halt as a result of COVID, same as it did for everybody else. And now the thing I'm doing is an annual conference in Dublin, in Ireland. The upcoming one will be on August the 17th through the 20th. I believe the website is actually going live today. And we have an absolutely stellar lineup of speakers. The distinctive things about my conference, first of all, it's very wide-ranging in terms of topic across the whole of aging and even things that might be considered somewhat tangential to aging. So it's exciting for the speakers because they will often learn stuff. Now, like most of the other speakers will be talking about stuff they don't know anything about, and they like that. But also, one thing about my conferences is that we very much emphasize the recreational aspect. We make sure that there's plenty of time for informal interactions between the speakers and the delegates. And perhaps largely for that reason, the conferences I've run over the past 20 years have been extremely successful in bringing new talent into the field, including people that were not working in aging at all before. I can't count the number of people that have come in of all ages, especially youngsters. And so that continues to happen. The audience tends to be quite knowledgeable, but a lot of them are, as you say, lay people. So the talks are, of course, appropriately pitched in that regard. And so, yes, absolutely, I hope that people will come to it. But there are plenty of other conferences. Some of them are more industry focused. I mean, there's some industry stuff, private sector stuff in my Dublin conferences, but it's not mostly that. Some that are more industry focused. Some that are more transhumanism focused, things involving things like nanotechnology that are less highlighted within the longevity community. There's lots and lots.
So, what steps can we take to do a more interdisciplinary science in the longevity field? You mentioned that there is no real path to do it because no one has done it before by definition, the interdisciplinary connections. How do you foster these connections?
So my conferences have been very effective in that regard. If you bring people together who have expertise in different areas and there is some potential for those areas to dovetail, then having them see each other's talks is the first step. And so I've been pretty effective in making that happen. And honestly, I don't know a better way than simply having a whole bunch of interdisciplinary conferences. Of course, I do a bunch of networking myself. I introduce people to each other when I see a synergy between different things. And sometimes it'll just be a case of me giving a talk somewhere myself and saying, these two people should work together, and sometimes they actually do, right? More or less, I'm oversimplifying, of course, but but yeah. So that's really what it's about. It's about the personal interaction comes first.
So, Aubrey, thank you very much for your time, but I would like to ask you one final question.
Okay.
What is the best advice that you have ever received in your career?
Yeah, it's a tricky one. I mean, okay, let me give a slightly indirect answer. So this was kind of before I had a career, when I was a teenager. My godfather said to me that the most important thing when you talk to someone that you should listen to is not what they say, but what they don't say. And one thing that I have found a little bit frustrating over the years is that I don't work that way myself. You know, I people often look for what I'm not saying and try to listen to that, and I try to stop them from doing that because I don't tend to write between my lines. Um, and I persistently find that people are reading things between my lines that I don't mean. But yeah, I think, finding the right balance there, both in terms of listening and in terms of what one says, is probably the single biggest thing.
Thank you for that advice. So Aubrey, thank you very much for your time. I really enjoyed our conversation,
Likewise.
and best of luck with your new foundation, with your
Thank
new
you.
events,
Yeah,
and thank you very much, Aubrey.
that's right. Thank you very much for having me.
Hello again. If you found this video interesting, please consider liking it and subscribing below. And also visit our website, quadrascope.com, to read about interesting news in the longevity industry.